So while continuing studies on mammary gland development, you worked and published on the effect of p53 mutants, carcinogen, CEBP, LIP, and their effect on mammary cancer. What triggered your interest on mammary cancer originally? Because you started with leukemia, lymphoma, and then...
So we were always interested in developing good models. The carcinogen model had one flaw, and that is you gave a carcinogen, you waited months, and then you ended up getting a tumor. But what happened in between other than a RAS mutation, we had no idea. And so it wasn't amenable to genetics. So in 1982, because we didn't have good cell lines to study casein expression, Kuo-Fen Lee, a graduate student in my lab, started making transgenic mice with Franco De Mayo. And this got us interested in genetically engineered mouse models. Kuo-Fen ended up going to Rudy Janish's lab and is now a full professor at the Salk, so he's done extremely well. But when he started it, he actually came into my office and said, "This is not going to work, I'll never get my PhD." So it wasn't clear back then that this was actually going to work. And it took Franco well over a year and more than 1,000 embryo transfers till we got our first mice. But it worked, and we could actually show a piece of the gene would work. So in about that time, in the early 80s, Team and Square happened, and I had a postdoc applicant who applied from China, and he came to the lab, and his name was Baolin Li, and he was interested in making transgenics to make some breast cancer models. And Larry Donhauer, a colleague at Baylor, had actually knocked out the p53 gene, one of the first tumor suppressors to be knocked out, but those mice were never getting lymphomas or breast cancer. They were getting lymphomas and osteosarcoma. So we looked through the literature at that time, and we realized that, in fact, in many cancers, they were starting to see mutations in p53. There was actually a small database from a group in Paris, from Soucy, and we looked at it and saw that there was an arginine to histidine mutation at amino acid 175, and also arginine to leucine or proline. And so we collaborated with Gigi Lozano, who had just come across the street. We got a construct, and we made these mutant p53, and then Baolin was able to actually make transgenic mice with them. We found actually that the different mutants, as Gigi had found subsequently, gave different effects. One of them actually induced apoptosis, but the other one actually that we were studying was a dominant negative or gain of function mutant. And we used that one actually in the mice, and by itself we didn't see tumors very frequently. It took well more than a year. But when we treated them with the DMBA, we got an acceleration, and that became a nice model. And then we collaborated with a postdoc, Daryl Hadsel, who had made an IGF-1 overexpressing mice, and again saw cooperativity. And then we got some mice from David Lee that expressed TGF-alpha, and then again. So it became a nice model. We eventually collaborated with Bill Muller and Arch Perkins at Yale and crossed it with an ERB2 mouse, actually, and that was a very nice. And these were aneuploid tumors, and it became a very good model. Now this was all based on really Phil Leader's original oncomouse, where you take MMTV-MYC, and that was the first transgenic model. And so, but we wanted to now model this more like human breast cancer. So and it turns out the mutant we chose is about 6% of all the mutations in breast cancer. It's one of the hotspot mutations. So that was really nice, and the other studies were really based on development. So in the casein promoter, there were not only STAT5 binding sites, but there were binding sites for CBP beta, as well as other transcription factors. And Brian Rott, a very good graduate student, was doing normal development studies, and he noticed different isoforms of the CBP beta, and they were changing in development. And we did some studies with Dan, again, looking at cell lines, and as you went from hyperplastic to tumors, you got more of the LIP isoform expressed. And that actually was a really nice paper in Cancer Research, and we collaborated with Nachum Sonnenberg and showed that this was being translationally regulated. Again, one of our interests, which we've kept many years. And so we took and looked in human breast cancer samples. It was our first attempt. Cindy Zano came as a postdoc from UNC. We were able to get 40 frozen tumors that we could do Western blots, because we didn't have antibodies to distinguish this. And when we did that, we found that LIP was overexpressed in aggressive ER-negative, PR-negative breast cancers. So we also made a transgenic model to study that. And again, many years later, Jean Massaguet in his lab showed that, in fact, the switch of TGF-beta from being cytostatic to promoting metastasis seemed to involve the switch in these CBP-beta isoforms. So it's still an exciting area. It turns out, even now in the lab, CBP-beta can regulate STAT3. It actually chips on the STAT3 promoter, so we're quite interested in understanding how it works in stem cells. Yeah. And so I would say that starting with the casein promoter, all the factors that we studied that bound to that promoter, eventually we studied in development looking at knockout mice, and eventually all of them moved into breast cancer. So I've always said that we have to understand normal development first, because that's really the basis of all these studies.
So more recently you also published a very nice paper on the P53 null mouse model and its effect on mammary gland generating different primary allografts. Can you tell us more about this model and about the different allografts that you generated?
Right. So again, this was a work that Joe Jerry had started when he had been a postdoc, and he worked with Dan Medina and Jenna Butel, and we were good friends. And we were always sort of concerned, why were we not getting mammary tumors in the P53 null mice? With this mutant P53, we clearly were able to.
Sure.
And so Dan and Joe realized that studies at Oak Ridge had shown that there were big differences in susceptibility of different mouse strains. And this was just at the beginning of a period where people were starting to really do mouse genetics in cancer. And that there were modifiers. It started by Bill Dove, actually, looking at the APC gene in colon cancer. And so Joe actually bred the P53 null mice into the BALB-C background and started to see some mammary adenocarcinomas, but they were still dying from lymphomas and osteosarcomas. So Dan and Francis Cottrell really pioneered the idea that you could transplant these null cells into a recipient, and actually these would give rise stochastically to the different breast cancers. Now Dan had done a little bit of work characterizing them, but he was mostly interested in preneoplastic progression and lesions. And so we were part of this Mouse Models of Human Cancer Consortium, and we were one of the first groups, Dan and I, to join this. And one of the goals was to try to characterize better these preclinical models. And so we sent some of the tumors to Chuck Peru, and there was a student in his lab, Jason Hershkowitz, and he started profiling these and could show that some of them clustered with a unique subtype called Cloud and Lobe. And I was lucky enough to be at an AACR meeting in Boston and met Jason. He had a poster, and I said, this comparative oncogenomics is where we need to be. And I recruited him back to our laboratory because we were also working on stem cells. And so with Dan and Francis, we started collecting now a very large group of tumors. In addition, I had a postdoc, Mei Zhang, who was interested in stem cells. And the idea of having a transplantable model was very attractive, because once we got tumors, we could make tumor banks and use them. To do stem cell work, you really can't wait for stochastic tumors to come up and get enough tumors to really do FACS sorting, and we had to have a model where we could set up a number of mice and actually do this. And so again, May worked with Francis and was able to do this for about 10 of the basal-like tumors, and do the first studies really in a syngeneic model of cancer stem cells.
We will come back to this. So this was like P53 null model, and now with the TCGA and other sequencing of human tumors, we know that this is one of the most frequent alterations in human breast cancer with PIK3CA mutations.
Yes, those are the two. Clearly the problem has been that P53 is not a druggable mutation, so we've known this for a long time. It's in the fact that 80% of the basal-like cancers have P53 alterations, I think, argues that it's really the driver in these types of tumors.
In 2000, you published on the CEBP control of cell fate determination during mammary gland development. What did we know about mammary gland cell fate back then, in 2000?
We didn't know a lot. I had a wonderful student, Tiffany Seagroves, and we were able to study the knockout mice. And when we started to characterize these, we realized that we were seeing epidermal markers. We knew, of course, from very classic studies that mammary gland developed from epidermal cells by invagination. And so we became very interested in that, and we actually followed work by John Wozlmerski and some other very nice work suggesting that in fact these cells could revert to an epidermal fate. And so when we saw these epidermal markers, we realized that something had changed in terms of the cell fate. In cancer we had actually seen this, because we'd seen squamous differentiation in certain tumors where we were making these keratin whorls with keratin 1 and keratin 10. So we had seen this occasionally even in the DMBA model, but at the time it was just sort of the beginning of the idea that there was sort of cell lineages and a cell fate determination, and what was regulating this we didn't really understand.
Then followed your papers on SCA1 positive cells, site population studies. Now, a few years later, how do you look at these years, and what did you learn from this site
I had another very good student, Brian Whelm, who came, and
who's now in Utah.
He has his own laboratory at the Huntsman, and we actually were a bit tired of breeding different alleles of knockout mice together, and we decided what we wanted to have is a system where we could take viruses and infect mammary cells and actually transplant them back in to look at gain of function and loss of function. And actually Trevor Dale and Paul Edwards had done studies like this with Maloney-based viruses. But it was very inefficient, and it only worked when you had a strong driver, an oncogene, that you could actually see a phenotype. But Trevor was smart enough to put a LAC-Z marker in so he could at least see he infected the cells. And so Brian started doing some of these studies, but we realized at the time we didn't know anything about stem cells in the mammary gland, other than you could transplant and there were stem cells. And we had a new colleague who came from Boston named Peggy Goodell, who worked on hematopoietic stem cells. And we also heard a talk from an epidermal biologist who was doing label retention studies. And so Brian got the idea that, based on having heard these talks, that we should look for a label retention population, and we should use some of the markers that were being used in hematopoietic stem cells. And so we used side population and SCA. We were also lucky that there was a SCA GFP knock-in mouse that had been made at WashU by Tim Graubert. We got those mice, and actually the terminal end buds were bright green, so we were very excited and said, and then we did actually studies with those GFP mice in transplantation to show that they would actually give rise to outgrowth. Probably it was a progenitor marker that still had limited outgrowth potential. We did not do serial transplantation back then, which these days we would have done. But back then Brian wanted to graduate and go do a postdoc with Xenoworm. And in fact it took Brian another 5 or 6 years to optimize lentiviral transduction of stem cells, which he then published in Cell Stem Cell. But was actually an appendix in his thesis in my laboratory. And so I think SCA was useful, but it clearly wasn't a marker, and we always said stem/progenitors because we never could prove. There was an enrichment in label retention in those cells, though. More recently, just a few weeks ago, Greg Hannon went back using a different approach to look for these label retention cells. And again, they are enriched in the stem cell population, but that clearly was not the appropriate markers. But starting with Mike Clark and the demonstration of in pleural fusions of a cancer stem cell, the field exploded. And then lots of investigators started looking at this. Perhaps John Stengel was one of the earliest who was trying to, again, coming from the hematopoietic stem cell background, to use fact sorting and look for markers.
So you also published on FGFR, on the pregnancy-induced non-coding RNA, the pink, on P190 row gap, and on the effect of other molecules in mammary gland development and or cancer. We will not go through all your over 180 or 170 papers, but I would like to discuss one other area of research from your lab, the one on cancer stem cells. How do you define this cell population to start with? I think there is a lot of controversy that is mostly due to semantic issues, so maybe we should start by telling us your definition.
So we, again, because we could do studies with transplantation in the mouse,
Yes.
wanted models where we could do functional assays to actually transplant in syngeneic models. And we knew we had a good model to do this. And so after Mike Clark's study, we started collaborating with clinicians to look at this in clinical samples.
So you are referring to Al-Hajj Pinaye's
Yes,
paper?
yeah, in
which
2003. And then Gabriel Dantou had a paper on mammoth
from
cirrhosis.
Max Wisha's lab.
For Max Witches. So again, the field exploded. The next year, Peter Dirks had a paper on glioblastoma,
Yes.
and so it was an exciting time. There had been a seminal sort of review by Tanisha Ray and Sean Morris and Mike Clark and
Yes.
Weissman in 2001.
Yes.
But what we thought we could do was actually not use primary cell lines or human tumors in immunocompromised animals, but we could take advantage of our syngeneic models. So Mei Zhang, I said, did a really beautiful piece of work where she could use the markers that Jane Wiesvader and Jeff and John Stingle and Connie Eves had developed in our mouse models, and then do limiting dilution transplantation in these syngeneic
Yes.
animals. And so we initially called these tumor initiating cells. I think Jeremy Rich likes to call them tumor propagating cells. But the idea is we didn't call them cancer stem cells because they clearly weren't just rare and quiescent cells, which is what people were initially thinking. And in fact, the definition in 2006 of a cancer stem cell is only a cell that self-renews and gives rise to appropriate progeny.
Yes.
And so we did, I think, all the appropriate studies to show this, and had a very nice paper, which May published in Cancer Research. And so this was the beginning of those studies, but at the same time I had a radiation oncologist who
Yes.
came over, who was an MD PhD, and had been out of the laboratory, and she wanted to work on Wnt signaling because she had done her thesis on that. And so we started looking at the effects of radiation on these cells and how the Wnt pathway influenced that. Again, and first initially in cell culture, and then later on in the mouse models. And again, that got us more involved clinically in the response to chemotherapy and DNA damage. At the same time, we were working in the clinic with Jenny Chang and saw
So this was in 2008 where you published with her.
Yeah,
the
it actually
GNCI
started,
paper.
so in 2005 we wrote a joint grant with Max Witsche and Rob Clark, actually. So all of us knew each other, we were excited about this field. We sent this as a DoD grant, and it was called the Concept Award, and we didn't get funded, even though about that same time Bob Weinberg said this is the single most important advance in the last decade in cancer. We quoted Bob, but that didn't help. We still didn't get funded. The review panel clearly didn't appreciate the importance of this.
The relevance of.
It actually led to, though, initial collaborations with Rob and people in Manchester, as well as Max and people in Michigan. And so I think, in many respects, even though we weren't funded, it was very important
to get together.
to get everyone together. And I think we actually complemented, and it actually led to clinical trials on Notch inhibitors later on between Jenny Chang and Max.
So in the paper the GNCI paper was entitled, or is entitled, Intrinsic Resistance of Tumorigenic Breast Cancer Cells to Chemotherapy. And later you also published another one in 2009, a Pinaye's paper, where this is the first demonstration that resistant cells from human breast cancer have features of tumor-initiating cells?
Yeah, it was the first demonstration in clinical samples. So some people had started to do some studies in cell lines showing you could generate resistant cell lines, but this was really the first clinical demonstration at the time, yes. And got quite a bit of publicity because it was done in patients.
So Jeff, you mentored many students and many postdocs. Many of them are now faculty in different places all around the world. How important was this for you?
This was the most important thing. I mean, to me, the success of my students and fellows was actually more important than the papers that we were publishing and the number of citations and the usual criteria people are using these days. I was really proud of how well my students had done. Almost every single one was in academics. I had very few actually that went into industry, and they all developed very nicely independent programs not always working in breast cancer, usually in other
Sure,
other
other
areas,
fields,
other fields. I'm most proud of that. I actually have several pictures from Gordon conferences with all my trainees there that
Yes.
I think is really the highlight of my career.
Actually, your website, the website of your lab, you can see all the pictures of
Right.
people over the years. So, Jeff, the French philosopher Michel Montaigne said, "Teaching is not about filling a vase; it's about igniting fires." Can you comment on this?
Well, I always felt that I could teach students how to do good science, but in fact, they had to come with a spark or enthusiasm. And so what I looked for in my students was an excitement about research to start with. I don't think I gave them that spark, but I definitely taught them how to do science properly, and then it self-reinforces itself. So I'm not sure you can light a fire under someone who doesn't have that spark
Sure.
to start with. But I do think that if they're excited about it and you're successful, and give them, again, the freedom to develop ideas, which I think is the most important thing, rather than just be a super technician,
Sure.
I think then it's very rewarding to see.
So, Jeff, what would you advise a junior faculty who is going to start her or his team to work on mammary gland development and cancer?
Well, I think you have to, I used to tell people you have to pick something that's exciting but not too exciting. So Stat5 was a good example. When we worked on Stat5 with just a few groups and it was just prolactin, it was fine. But then every cytokine was regulating Stat5, all the immunologists became interested and the field exploded. And so at that point I decided to move more into development and do other things. I think you have to pick something that is a unique question that you can study. But you have to be careful, because I had some very smart friends who picked really exciting questions, but they couldn't get answers in 3 or 4 years, therefore they didn't get funded and,
Yeah, sure.
and they didn't survive. I think you have to pick something that is really, you're asking a question hopefully that not a lot of people are asking, and that's the key. At the same time, have a few things that are guaranteed to work so that you can continue to...
Sure, some low-hanging fruits.
Yeah, low-hanging fruit, so you get some publications and you can continue to get funded.
So, Jeff, what are the major questions that you think still need to be answered in the field of mammary gland development?
Well, I think there's still a lot to be learned on the cellular hierarchy in mammary gland development. I personally think that the cell-cell interactions and both paracrine and autocrine pathways,
That's the niche.
and the niche, but also just even between luminal cells and basal cells, and understanding the kind of crosstalk that's going on is really. But we have to understand this as a sort of cell biology system, not just as a single cell with a receptor. And so that's, from a developmental point of view, that's really the next stage. We have to do this. One of my famous quotes is that the mammary gland is really the Drosophila eye of mammalian functional genomics. We have to do it like the Drosophila geneticists have done this, where we have a patch of cells that are knocked out, surrounded by wild-type cells, and understand in these chimeras how things are interacting. I think that's the big challenge right now. We can do that. We have the genetic tools. We can do lineage tracing now, so we have to do lineage tracing. So it's an exciting time. I think we're going to be in log phase very soon in terms of these kinds of studies.
But ultimately, we would like to know about the human breast. And the model is, like one of the colleagues said, a model is a lie that tells us the truth. So ultimately, we need to go from the mouse to the human.
Right.
This is probably also challenging.
It's very challenging. I mean, I think there are some considerable differences, especially in the stroma,
Yes.
that we can't model particularly well. We have to actually do this by doing studies in immunocompromised animals, so that there are limitations. Eventually, we're going to have to humanize the mouse, so that we can study things like IL-6 in the mouse and human, and prolactin in the mouse and human.
Yeah. So, what are then the major questions that you think need to be addressed in breast cancer?
Well, I think the clinical questions. It's not only in breast cancer, but most cancers these days are really the escape mechanisms or resistance mechanisms and bypass for therapy, what actually regulates dormancy and recurrence, and how can we really clinically address these two key issues. If we can keep cells dormant, patients most likely will die from something else.
Sure.
I never felt we were going to cure cancer. I think we can manage cancer.
Yes.
We need to be able to do that.
So people will not die from cancer, but die with
I think
cancer.
we have to understand cooperative pathways and how we can target
Sure.
with multiple pathways, because single pathway agents are going to always fail.
So what's about the metastasis question?
Well, I think that's we haven't done a very good job modeling metastasis in terms of treatment and testing, especially in terms of developing drug treatments. What we've done traditionally is actually treat primary tumors.
Sure.
It's clear that the environment changes the response to treatment. We have to actually start doing this in models, better preclinical models, to look at metastasis. And really, I think, again, have to change our endpoints, so we're not just looking at the shrinkage of the tumor, but actually what the cell populations are and what's going on.
And now from sequencing data, we realize that there are many mutations that are not frequently found, but they are there. And whether these mutations have any functional role is not known.
I think it's a big unknown. We have to do functional genomics at this point. We can't just do descriptive genomics. So, and that can't be done with making knockout mice for every gene.
Sure.
That's one reason I think some of these lentiviral approaches may actually be more robust and allow us to study this.
So there are a lot of exciting projects for the next generation.
Yes, there's more than enough.
More than enough. So what would you then like to tell the next generation of scientists who will watch this video?
I think that you have to have the spark, the enthusiasm to start with. You have to be excited. If you're
curiosity.
If you're not really excited about this type of field and research, you shouldn't be doing it.
Sure.
That's the first thing. If you are, I think you will see, as I've seen, actually significant changes in the response and treatment over the years. I mean, there's been a 25% reduction in breast cancer mortality during the time that I've been studying this. I saw children who used to die in 3 to 6 months that were living, 5, 10, 20 years with other problems, but still, I mean, remarkable successes.
In leukemia.
In leukemia. So I think that if that doesn't inspire you, you should be doing something else.
Yeah. So Jeff, is there any question that I should have asked but I didn't? I know that there are many, or possibly, but is there any particular one that you would like to...
One of the fun things that we got into as sort of a side project over the years was the biotechnology of and we were able to actually make designer milk. We could target different proteins to be expressed in milk. We actually trying to make surfactant protein to treat respiratory distress syndrome. We made follicle stimulating hormone. And this was actually quite fun because we got to interact with both some large companies like gene farming, as well as really go to different kinds of meetings and meet a totally different audience. It even got me to Cuba as a guest speaker. So I think you have to be, I think, flexible and when you have these opportunities, take advantage of them. And that's really the only thing. The other thing is give the people who work in your group flexibility to develop their own projects outside what you're working on. And then that's how we developed the prostate cancer model. In fact, my most highly cited paper is actually on the development of the TRAMP model, not on anything we've done in breast cancer.
So Jeff, thank you very much.
You're welcome.
It was a pleasure.
It's been a pleasure doing this. Yes.
Likewise.